Hello everyone. Today we will be seeing a video on biological database. I'm Dr. Anand Tyagaraj. I'll be taking this lecture today. So before going into the topic, I would like to show you some databases in molecular biology. So many would have seen many of these databases who are in molecular biology like DB, ST, COPS, CATH, but if you happen to see the list that I have put here, it's a very huge list and it is still keeping on increasing. I've tried to comprehend all the information and try to show here. Before going, so there are key features that these databases have. Let's see about these features. Most important feature is comprehensiveness. Then comes accuracy and reliability. Then these databases should be user-friendly uh, with user-friendly interface, and they should be regularly updated. That is on daily basis, and the data should be stored in a real-time manner. Then comes interoperability. That is, it should be compatible with other databases and bioinformatics tools. Data accessibility. It should have open access or subscription based with clear policies. And apart from it, the databases that we are using today in molecular biology have query access and analysis tools. So, for most popular analysis tool like BLAST from HCBI are very popularly used. Then, because all these databases more or less deal with the similar kind of data, that is DNA, proteins, uh, these data should be having common formats like FASTA, PDB, XML for seamless integration. Apart from this, the data should be in a proper manner for visualization, like it should be represented as graphs, pathways, or 3D structures to get good insights. Now that we have understood all the biological features, let's try to see the classification of biological databases. Broadly, biological databases are classified as primary, secondary, and composite. So, the key feature of primary databases are the databases where the data submission is enabled. That is, your data from your research is submitted to these databases. So, for example, if you happen to generate a DNA sequencing data, then you can submit to a nucleotide database, and this is a primary database. Similarly, if you have an extracrystallography structure, then you can submit it to database like PDB. Or if you have a protein sequence, then you can submit it to UniProt. So, or also literature like PubMed is a popular database where submission is possible. Secondary databases are those, the primary data are integrated. So, here you have data. So, upon integrating, you will end up having information which is stored in the secondary database. When you are doing this, the data is very well curated and it is stored for more insights into the data. So, combining secondary database features and some more data into it and obtaining insight into it, you will end up having what is called as a integrated database, which is nothing but composite database. So here are some based on data types. This is the broad classification of biological database. Apart from this, we can also classify biological database based on data type, based on accessibility, based on data source, and based on curation. So as I said, based on data type, it's sequence data, structure data, pathway data, and interaction data. Based on accessibility, primary, secondary, composite, which we saw in great detail. Then based on data source, you can classify as genomic data, proteomic data, and metabolic data. And based on curation, manually curated or automatically curated. And today's AI generation, more of automated databases are in the rise. Next, we will try to see one by one each and every databases. First, we will be seeing sequence databases. So, under sequence databases, you have broadly classified as nucleotide database and protein database. So, here are the popular nucleotide database that we all use on everyday basis, like EMBL, NCBI, that is GenBank, then DDBJ. All three form a consortium, and then you have nucleic acid database, then DBSNP is one popular database, and then genome sequence archive, GSA, we use. So coming to protein, you can see most popular ones are PIR, Protein Information Resource, SWISSPROT, then you have PDB, then CAT, SCOPE, MINT, and some more databases, popular database like KEGG. So coming to nucleotide database, nucleotide database, as name suggests, is a repository for storing DNA, RNA sequences from various organisms. They contain raw sequences, annotations, and related metadata, and they are used in genomics, phylogenetics, and molecular biology research. So a few key nucleotide databases are GenBank, EMBL EBI, DDBJ, and RefSequence. Key features of these nucleotide databases are they store raw annotated data and assemble sequences. They enable features like blast searches for sequence alignment, then support comprehensive genomic and evolutionary study. Apart from it, provide FASTA and GenBank format which can be easily retrievable. Let's see some standard contents of sequence databases in the coming slide. So usually the sequence data would contain raw sequence or protein sequence in standard format like FASTA and GenBank. Then you will have basic annotations of these data like gene name, function, location in the chromosome, coding region or non-coding region, and what is the protein coding potential. Apart from it, you will also have metadata like organism name, taxonomy, strain information, then source details like genomic, transcriptomic, and synthetic origin. So each and every data here is going to have what is called as a unique identifier or accession number. So for example, a GenBank accession number will look something like this, NM_0012567889. So this enables easy access of unique data or specific data because of the unique identifier. Apart from this, you also have reference and citations associated with the work, then publication and submitted details associated with the sequence, then cross-references, links to other database like PDB, structures, KEGG pathways, UniProt, and proteins. Apart from this, you also have sequence features like exon, intron, promoter, and regulatory elements. Now let's see one by one different databases. First, we will be seeing NCBI. NCBI, full form is National Center for Biotechnology Information. It's a key bioinformatics resource under National Library of Medicine, LLM, USA. It provides access to vast collection of biological data for genomics, proteomics, and literature. And it supports research in molecular biology, genetics, and computational biology. So this is the site where you can access the NCBI, that is www.ncbi.nlm.nih.gov. Now let's see what are the possibilities in NCBI. So sequence submission is one of the key features of NCBI. You have this is done by using Bankit or Sequin, and then you also have a search engine for data retrieval, which is Entrance, and this receives information across all resources under NCBI. Example: PubMed, Taxonomy, SNP, and PubChem searches, etc. So there are also other key databases under NCBI like GenBank, PubMed, which is very widely used, BLAST, which is a tool for doing sequence and pairwise sequence alignment, then RefSeq, which is curated reference sequence for genes, proteins, and genomes, SRA. Today we are in a NGS era where SRA is the most popularly used database. Then protein database like stores protein and annotations, then DBSNP and CleanVar contain genetic variation and clinical significance. Let's see some various features of this NCBI. Key features are BLAST, that is basic local alignment search tool, then primer BLAST, where you can, for a given sequence, it designs primers and give you, then B-Link, then ORF finder, ORF is open reading frame finder, then genome workbench. Other features are like they are freely and publicly accessible, comprehensive biological data storage and retrieval, and it supports sequence alignment, genome annotation, and comparative genomics. Then it is integrated with multiple bioinformatics tools and resources. Now that we have seen one primary nucleotide database, let's try to see a protein sequence database in the coming slide. So, a protein sequence database usually stores protein sequences, structures, and functional annotations. So, this is used for studying protein function, structure prediction, and evolutionary relationship. And they also support bioinformatics analysis like sequence alignment and homology modeling. Few key protein databases are UniProt, where protein sequence is the primary data there and also has functional information about the sequence. SysProt, this is a manually curated high quality protein sequence. Then TREMBL, which is an automatically annotated protein sequence database. Protein information resource, it's an integrated protein sequence database, very comprehensive database. Then protein data bank, which has all the 3D structure of protein. Then PFAM is a collection of protein families and functional domains. So some key features are like stores primary and derived protein sequences, provides annotations on functions, domains, and pathways, supports BLAST search for query similarity, used in drug discovery, structural biology, and functional genomics. Coming to UniProt, it's a comprehensive protein sequence for functional information. It's a functional information database. It integrates protein sequence from various sources, supports research in proteomics, drug discovery, and structural biology. The key components in UniProt are SysProt, TREMBL, UniRef, and UniPrac. Some key features of UniProt are provides protein sequences, structures, and functional annotations. Then it includes protein-protein interaction, pathways, and post-translational modifications, supports sequence alignment and homology modeling, and integrates PDB, KEGG, and other bioinformatics tools. Next, we will see protein family or PFAM. This is a comprehensive database for protein families, domains, and motifs. This uses an algorithm called as hidden Markov model to identify conserved protein domains. It helps in functional annotations and evolutionary studies. Key features of PFAM are they are protein family classifications. It has HMM and it uses cross references and also gives graphical representations. So, protein family classification is based on conserved functional domains between the proteins. So, it is widely applied in protein function prediction on conserved domains, structural and evolutionary studies of protein families, identification of novel protein function, and supports of genomic annotation. Now that we have seen all the sequence database, now let's move to structural database. The most popularly used structural database are PDB, CATH, and SCOP. PDB stands for Protein Data Bank, CATH stands for Class Architecture, Topology, Homology, and SCOP stands for Structural Classification of Proteins. Let's see PDB first. So, Protein Data Bank is a global archive of 3D structure of biomolecules such as proteins, DNA, and RNA. Although its name says Protein Data Bank, now the database has 3D structure of even DNA and RNA into it. It serves as crucial resource for structural biology and drug discovery. The key features are it stores X-ray crystallography, NMR, and cryo-EM derived structures. This provides atomic level details of biomolecules, supports molecular modeling and drug design, and open access and maintained by WWPDB. This is worldwide PDB. So major resources are RSCB PDB USA, PDB-E Europe, and PDB-J Japan bioinformatics. So all these are integrated and maintained by WWPDB. Major applications include drug discovery, molecular modeling, and structural biology. You can access PDB from www.rscb.org, where you can unlock the molecular world one structure at a time. SCOP is, as I said, it's structural classification of protein. It's a secondary database, secondary structural database, where the protein structure data is it's a hierarchical database that classifies protein structure based on evolutionary and structural relationship. It provides insight into protein fold, superfamilies, and domains. Key features include classifying protein into families and superfamilies and folds based on evolutionary relationship and 3D structure, derived from protein data bank entries, aids in protein function prediction. SCOP classification levels include class, fold, superfamily, and family. And application of SCOP include protein evolution, drug discovery, and bioinformatics. you can access scope from this particular database. Now let's move to other structural database called as KEGG. KEGG stands for Class Architecture, Topology, Homology. It's a protein structure classification database. Again, it also does hierarchical classification of protein structure, grouping them into structural and evolutionary relationship. It helps in understanding protein function, evolution, and drug discovery. Key features include classification of protein domains based on structure and function, uses automated and manual curation, derived from PDB structure, facilitates functional annotations and evolutionary studies. Application again involves structural bioinformatics, drug discovery, evolutionary studies, etc. So this is coming to the end of biological database. Thank you for patient listening. Thank you.